Hello, and welcome to Chapter 14 on DNA, Genetic Material of the Cell. This is our final week of the semester. Congrats on making it and persevering this far. Your final mini exam will cover this chapter, Chapter 14, and the following Chapter 15 on genes. The objectives for this chapter are to know the structure of DNA. I won't expect you to be able to draw the DNA structure by hand, but you should be able to identify the components within it and be able to predict how its strands would pair with each other. Second, you should be able to understand why replication of DNA is called semi-conservative. So what does that mean? There's an experiment, a classic experiment that we'll talk about that determined this, and you should be able to understand and interpret the results of that experiment. Third, you should know the general mechanism of how DNA is copied, and we're going to learn it first in prokaryotes, and then we will learn about how it works in eukaryotes, because it's a little bit more simple in prokaryotes. And then finally, we're going to talk about DNA repair, and you should be able to describe or compare and contrast two types of DNA repair. So first, let's talk about the general structure of DNA. So nucleic acids include DNA and RNA, and DNA stands for deoxyribonucleic acid. Right? So what does it mean by deoxyribo? Right? So ribo has to do with ribose or sugar. So if you look at an individual nucleotide, so one base in a sequence of DNA, you can see that we have this in blue, this ribose or sugar molecule, and it has been attached to a phosphate group, and we call this in pink on the top right a nitrogenous base. Right? It's called nitrogenous because you can see there's lots of nitrogens in it. Now, the difference, or one of the differences between DNA and RNA is what is attached to this sugar, right? So in this one spot here, you could have just a hydrogen, or you could have a hydroxyl group, or OH, right? Oxygen and a hydrogen. And so if it does not have this hydroxyl group, it's called deoxy, right? Deoxyribonucleic acid. So again, in ribonucleic acid, or RNA, it's going to have a hydroxyl, an OH group here, attached to the sugar, the ribose. And if it is DNA, deoxyribonucleic acid, it's going to be missing the hydroxyl group at that position. Again, I don't expect you to be able to draw this structure by memory, but you should be able to identify in this structure where the phosphate group is. Where is the nitrogenous base? Where is the ribose? Now the phosphate group, I'd like to point out which carbon it's attached to. If you were to count the carbons in this sugar, you can see we have 1, 2, 3, 4, 5 carbons on this, and attached to the 5th carbon is the phosphate group. I'd like you to also notice the properties of this phosphate group. See how there's negative charges on those oxygens? So this can impart a negative charge to DNA. Attached to the first carbon in the sugar, we've got our nitrogenous base, and the particular structure of this base can vary. You know that you've got a sequence of DNA, right? A, T, T, C, G, A. Those are representing the nitrogenous base, because everything else in this strand is going to stay consistent no matter what nucleotide you're looking at, except for this nitrogenous base. So you can see that adenine and guanine have a little bit different structures than cytosine, thymine, and uracil. So they're called purines. They have 2 of these ring structures, whereas the pyrimidines only have a single ring structure. So again, when you're looking at this structure here, the phosphate group and the ribose group when you're looking at, say, across a sequence of DNA, will not change at all. Just the nitrogenous base will change, and it can be any of these. Now we've got an exception here, uracil. Uracil is only found in RNA, right? And uracil replaces thymidine. Thymidine and made up a new nitrogenous base. So in RNA, uracil is taking the place of thymine, but today we're talking about DNA. Now you should be able to distinguish between purines and pyrimidines and know which bases are which. So adenine and guanine are both purines, and there's a couple ways you can remember this. So first, you can think about the length of the name and the size of the molecule. So purine is short in name, and the molecule is the opposite. It's really big, whereas pyrimidine is a big name, but the molecules are relatively small. Alternatively, you could remember cut the pyramid, cut C-U-T, cytosine, uracil, thymine, are pyrimid pyrimidines, and then the remainder, A and G, adenine and guanine, are the purines. So whatever you need to do, you should be able to just know. Again, you don't need to memorize these structures, but if I gave you them, you should be able to classify them into purines or pyrimidines. And if I asked you to list off all the purines, you should be able to know that they are adenine and guanine. Now, in a DNA strand, we have multiple nucleotides, right? It's a polymer, so it's got pieces that are put together. And so in this cartoon, we have two nucleotides that have been attached, right? It's been shortened. The drawing of the structure has been simplified. For example, this phosphate group before had more detail in it. It's just been simplified here, as well as this base, right? It's more generic looking just for simplicity's sake. And what we want to focus on is how do these two nucleotides connect? If you remember way back at the beginning of the semester, we talked about what type of reactions put molecules together. Do you remember? Condensation reactions, right? So a condensation reaction occurs to put these two subunits together, and the way they combine is at a phosphate group of one and the hydroxyl group of the other. Right, so again, this bond is called a phosphodiester bond. You should know the name of that bond, and you should be able to identify in a strand where those bonds are. Right, so again, before these were bound together, if you were to count 1, 2, 3 at this third carbon, there used to be a hydroxyl group. And on this one, 1, 2, 3, 4, 5, on the fifth carbon is the phosphate group. So the phosphate group of one molecule attached to the hydroxyl group of another. Now when we describe chains of DNA, or these polymers, we usually orient them in this 5' to 3' direction. And so you can see on this drawing, they've pointed out a 5' end and a 3' end. And we'll talk more about how to identify 5' and 3' end, but for now you can notice that the 5' end has the phosphate group attached to it at the very end, and at the very end of the 3' end is a hydroxyl group. I'd like to take a note just to talk about history of science. Rosalind Franklin is credited with helping to determine the structure of DNA, particularly that it is a helical structure. So far we've been looking at it as just kind of a straight strand, but it actually does have this winding helix shape. And she collaborated with some other scientists performing X-ray diffraction, and she found that the diameter of DNA is 2 nanometers and kind of the height of one complete turn of the helix. I don't expect you to memorize this data, I just wanted you to make a note because she often gets overlooked and did get overlooked in terms of recognition for this work. She worked with James Watson and Francis Crick, who you may have probably heard of before, they were the ones that determined kind of other details of, or additional important details of the DNA structure. And while while Rosalind did a lot of the like wet bench work, all of their work was very kind of theoretical, looking at information, looking at other people's experiments, and kind of working with space and orienting molecules. And so they were able to propose the double helix. So the fact that it's not just a single strand in a helix shape, but that it is two helices that are wound around each other. And that's what we can see in this cartoon on the right. The nitrogenous, the backbone of the phosphate groups, the sugars, is shown with this blue ribbon, and then the nitrogenous bases are shown kind of with the bars. So again, the sugar and the phosphates don't change between one base and the other, and they are what form up the backbone of the DNA. And so this backbone is sometimes called a phosphodiester or a sugar phosphate backbone. And we're just looking at one strand here, and I'd like you to take a note to identify the 5' end and the 3' end, and which end is which. Here we have the DNA represented as a double helix, and rather than having it twisted, as it normally is found on the right, we have in the middle where it's been untwisted. And I want to focus on kind of how these two strands interact. So in this, we've got our ribose sugars, deoxyribose, sorry, shown in blue. The phosphate group is simplified as just a P with a circle around it in yellow. And here we've got a little bit more detail on the different nitrogenous bases. Take a minute and see if you can figure out which of these are purines and which of these are pyrimidines, and see, do you notice any patterns? So here we have a pyrimidine paired with a purine. Here, a purine paired with a pyrimidine. So pyrimidines and purines pair with each other. Right? I'd like you to also notice the pattern. We've got T binding to A and G binding to C, and it is always that case. Right? A never binds to C, for example. So the complement is what we call the base that binds to this base. So the complement of thymine is adenine. The complement of guanine is cytosine. The last thing I want to point out as well is the type of bonds that we have here. So the bonds that put one nucleotide, attach them to another, those phosphodiester bonds, those are covalent bonds. What about these bonds down the center that are involved in the complementary pairing, right? As you can see, these are hydrogen bonds. Hydrogen bonds are non-covalent bonds, and if you remember from back at the beginning of the semester, that non-covalent bonds are not as strong. They can be temporary, but if you have a lot of them together, they can be quite strong. And so, depending on the temperature, you can actually separate DNA between double-stranded structure and single structure. At cooler temperatures, it is in a duplex form, but if you warm it up, you can break these hydrogen bonds and split the DNA into two strands, which is kind of neat. As you're studying, another thing I would encourage you to do with this structure is to try and figure out where exactly would you find the phosphodiester bond, say, between these two nucleotides? Right? Where would it be? Could you point to it with an arrow? Now, the last, I keep saying the last thing. The next thing I want to point out here about says anti-parallel strands. So what that means is that these strands are parallel, right? They're kind of lined up with each other. It's not like they're this or perpendicular. They're parallel to each other, but they are anti-parallel. So if you look on the top left, you can see that this end is the 3' end, and this end is the 5' end. However, the complementary strand over here is the 3' end, and the complementary end over here is 5', right? So the opposite ends pair with each other, and that's why it's called anti-parallel. Again, let's look a little closer at these nitrogenous bases. Here we've got guanine paired with cytosine and adenine paired with thymine, and you can see that hydrogen bonds are what are the bonds that mediate these pairings. And in one set we have 2 hydrogen bonds between A and T. We have 3 hydrogen bonds between guanine and cytosine. Let's talk about identifying 5' and 3' ends on a strand of DNA. Right? So I'm just going to draw one strand of DNA. And I'm just simplifying. I'm drawing the phosphate groups as just a P with a circle around it. All right, so here we have a simplified diagram of one strand. And just to kind of show you more up close what this would look like, here's just kind of a simplified drawing, or a more with all the details, and then here we have our simplified drawing. Now, this end of the DNA is called the 5' end, and this end is called the 3' end. We know that the 5' end has a phosphate group attached to it at the very most end, and that the 3' end has a hydroxyl group, also known as, right, this O and an H together. I'll just kind of highlight these. But why is it called 5' and 3'? What you have to do here is count carbons. Yay, right? Chemistry is the foundation for biology. So when we count carbons, we are going to start over here, right? Let's do it on our more complex drawing, right? 1, 2, 3, 4, 5. Those are the 5 carbons in our ribose. And so what you see here is that, oh, I did draw it on the wrong spot. I had it in the right spot to begin with. All right. That our hydroxyl group is 1, 2, 3. It's on the 3rd carbon in this ribose, hence why it is called the 3' end. And when we count where this phosphate group is attached here, 1, 2, 3, 4, 5, the phosphate group is attached to the 5th carbon in that sugar, right? So you should be able to point out why exactly it is that this end is called the 5' end and this end is called the 3' end. It is not sufficient to say this is the 5' end because it has a phosphate group attached to it. You have to know that it's called the 5' end because the last functional group, which is a phosphate, that's attached to this small ribose is attached to the 5th carbon. And here, the very last functional group attached to this ribose, the hydroxyl group, is attached to the 3rd carbon, and that's why that's called the 3' end. All right, so let's talk about why replication of DNA is semi-conservative. Now, when scientists were first trying to figure out how DNA replicates, they had three theories, and then there's this classical experiment we're going to go over that allowed them to determine how DNA is copied. So on the right here, we have three potential mechanisms by which DNA could be copied: conservative, semi-conservative, and disparate. And so you can see that in conservative, you have all the new DNA is in a complete helix of its own. In semi-conservative, you have in each duplex one new and one old strand. And in our last option, you have kind of a mixture within each strand of old and new DNA. So again, we've got the conservative model, semi-conservative model, and dispersive model. And so they found out that the way that DNA is replicated is following this semi-conservative model, so this one in the middle. The way that they did this experiment way back in the late 50s, Meselson and Stahl are famous for this, is that they needed to figure out a way to label old DNA versus new DNA. Right? So in that previous picture, we had old DNA in blue and new DNA in yellow. And so the way that they labeled DNA was by controlling what it was grown in, what it was made in. Right? So if you remember when we talked about the structure of DNA, we have these nitrogenous bases, A, T, C, and G, that have a lot of nitrogen in them. And so the scientists figured out a way to label that nitrogen. And so they have an isotope of nitrogen. We've talked about isotopes before. If you remember when we talked about radiocarbon dating, you can go back to that chapter if you want to figure out what isotopes mean. But they have one isotope called this heavy isotope, 15 nitrogen or nitrogen 15. And when DNA is made, these bacteria can actually pick up the nitrogen from their surroundings and use that to make those bases A, T, C, and G. All right. So now the nitrogens in those bases are labeled with this N15. And so they're going to call the original DNA heavy DNA. And then they took the cells out of that medium and put it into a different medium that contains a lighter isotope, 14N. And so any new DNA that would get made at this point, once it's switched, is going to have this 14N in the nitrogenous bases rather than this heavier 15N. Right? And so this allows them to identify or to distinguish original or old template DNA and the new DNA is by labeling the nitrogens. And because these isotopes have different weights, the DNA itself is actually going to be lighter when it's using the 14N and going to be heavier, the molecules are going to be heavier when they have the 15N isotope. And so if they have a mixture of DNA and they can sort it by weight and see what type of proportions, what is the composition of their media. Right? So again, they grew the cells for a period of time for several generations in this original medium that had nitrogen labeled with 15N. And then after all the DNA has been labeled with that, they switch the cells into a media that has this lighter 14N. And so any newly made DNA, any copies of DNA would be lighter in weight. So let's take a minute to really think about this experiment because I find it a little confusing unless I break it down. So here I've drawn two examples of nucleotides of DNA. One nucleotide is from the cells that were growing in the heavy nitrogen medium, right? So as this nucleotide is getting made, the nitrogen from the environment is getting put into the nitrogenous bases. And so this molecule of DNA is going to be, again, called our heavy DNA. This is our original DNA. And so then what they did was they took those cells and they exposed them to a new medium that had our lighter isotope of nitrogen. And so as any new DNA is made, the new DNA will incorporate this lighter isotope of nitrogen. So now you have a way to identify new DNA and distinguish it from old DNA, right? And this is physically lighter than this DNA. And so if you put this DNA into a tube and you centrifuged it with a special solution, you can separate out the DNA by weight. So this old DNA is going to show up lower on a vial that's been centrifuged because it's heavier, and the newer DNA is going to show up as lighter. And so by determining how many bands, how many layers of DNA result after centrifuging the cells, you can determine kind of what's going on in terms of the replication of the DNA. So let's talk about what possible results we could see. Right? So if you have DNA in which our conservative model, right? At this point, if you have all new DNA together and all old DNA together, what you're going to see is a light band and a heavy band. Right? Now, if you had a situation such as our semi-conservative model, in which you had both, in each duplex, you had one old and one new strand, you're going to see one band. It's kind of an intermediate weight, right? Kind of in the middle. The same thing is what you would see if you had the dispersive model, right? Where you've got kind of combinations, mixture of DNA. If it was kind of an even distribution of old and new within each strand, you would also see one intermediate band. So let's move on to the next slide and see what they found. So to really tease out what was going on in this experiment, they isolated DNA at multiple time points, all right? And so let's look at the results on the right here. So they collected cells after they had been growing in the new medium, first at 0 minutes, then at 20 minutes, and at 40 minutes. And so they know about how fast DNA, how fast the cells replicate. And so they knew that after 20 minutes, the cells had copied once. And so that would show one round of duplication. And after 40 minutes, that would show a second round of duplication. And so what they found is they collected those samples at each time point. And then on the bottom here, we have the results of their centrifugation, right? I don't know why the figure in your book has turned these tubes on its side, but the left is going to be higher up. A band that's higher up, more to the left, is going to indicate lighter, and a band that is to the right is indicating more to the bottom of the tube and that it's a heavier DNA. And so what they found initially, right, when no DNA replication has yet occurred, that we just have a big band, all the DNA is heavy, right? Because originally all the DNA was cultured in that heavy isotope. Then after one round of DNA, they found a band that was intermediate, right? So this could indicate either the semi-conservative model or the dispersive model, right? Because they could both show the same results. However, once the DNA had a chance to duplicate a second time, they found that they had two bands. They had an intermediate band, as they saw before, but also a band that was even lighter, right? And so what you see here is what they're hypothesizing what's happening is that during the first round of DNA replication, you see that you have the template, the old DNA is acting as a template for the new DNA, and then eventually that new DNA is being used to make more new DNA. And if we were using the dispersive model, no matter how many times the DNA replicated, you would just see a bunch of mixed up DNA. You would just see that one intermediate band. And so the fact that they saw a second band that was lighter in addition to that intermediate band really helped them prove that the semi-conservative model was in fact true and not the dispersive model. Again, here you can see we've got our three possible models, right? And with the conservative model, you would only see two bands, a top band and a bottom band, right? And that is never the case in any of these options. With our second option, you would at certain points, you would see an intermediate band, which they did, right? But you would also eventually see that the new DNA is being used as the template. And so we have an additional band here that is made up of all new DNA. However, if that was not the case, no matter how many rounds of cell division you had, you would have this mixture of DNA that was both heavy and light, and so you would only see one band in the middle. So again, the fact that after the second round of division we see a higher band in addition to this intermediate weight band tells us that it is semi-conservative. The fact that we see an intermediate band at all rules out the conservative model. Let's move on to our third objective, getting into more of the details of the mechanism of DNA replication and how it occurs in prokaryotes and eukaryotes. So I like to think about DNA replication as a photocopier, right? If you're going to use a photocopier, you need three things. You need something to copy. In this example, we have a sheet of paper that's being copied. You need something, a machine, to do the copying. And you need pieces of paper, right, to use as your copies. You need to have paper stocked in the machine. So the same thing happens in DNA replication. The something to copy is your parent or original DNA molecule. The thing doing the copying is an enzyme called a polymerase. And the building blocks to make these new copies are called nucleotide triphosphates. Now, the stages of DNA replication can be broken down into three, basically beginning, middle, and end. We have initiation. Replication begins. So all of the machinery has to be brought together in the nucleus for the DNA to copy. Second, we have elongation. So the DNA polymerase is running. We've got a new polymer, a new strand being made. Ultimately, all the DNA gets copied and we have to have termination. So those enzymes and the machinery leave the DNA. Now we said that the new material, the building blocks, are called nuclear triphosphates. So I'd like you to notice here, sorry, ouch, sorry, I'm trying to grab my mouse and I just grabbed my cactus on my table instead. So what I want you to notice is on the bottom left, we have our new incoming nucleotide. All right. It looks a lot like the rest of the nucleotides that are already in the DNA, but can you notice the difference? Here we have 3 phosphate groups. That's why it's called a nucleotide triphosphate. And when this new nucleotide gets incorporated into the growing strand, you can see that those 2 phosphates are lost. Again, the enzyme doing this work is called a DNA polymerase. I'd like you to note on which end is the new nucleotide being added. It is being added to the 3' end, as you can see here. Right? So DNA polymerase is functioning, it's making the new strand in the 5' to the 3' direction. And remember again, this is anti-parallel DNA, so the template strand, the original strand, the 3' end corresponds with the 5' end of the new strand. Now, let's talk more about this DNA polymerase. All right. So in order for a polymerase to work, all right, what it does is it finds complements, right, bases that are complementary to the bases in the DNA strand that exist, recruits them and helps to bind them and attach them. It first has to, an RNA polymerase first has to make a primer. And what this primer does is it's kind of like a flag that's telling the DNA polymerase, this is where you need to go, this is where you need to start. And so even though you're copying DNA, a small portion of it, this RNA primer, is temporarily there. So you've got RNA temporarily in the new strand. And then once the polymerase is done, that RNA gets removed in that primer area and replaced with DNA. Right? So first thing to note, for a DNA polymerase to function, it needs to have an RNA primer that's put down. Second, you can see it's making this new DNA in the 5' to 3' direction. Very important. Right? And what is it doing? It's bringing in those complementary bases and it is connecting them together, forming phosphodiester bonds. Remember we learned about those, to make this chain. Now in prokaryotes, this example we're looking at is in E. coli. They don't have a lot of DNA, and so their DNA is actually in a circular structure, and it's just one ring. And so what happens is on one end of this ring, we have the origin. Right? So this is where the initiation of replication begins. And you can see that we have this kind of like a bubble forming. So this enzyme called complex called a replosome will unwind or separate the DNA duplex. Because as you saw before, we need to get the enzymes, the DNA polymerase, to have access to the one strand, the template strand, so that it can read that sequence and determine what the complementary bases are that need to be brought in. So in this case, we have two complexes, two replisomes. They just go in opposite directions around this circle. When they both reach the other side, they let go. And you can see now that we've got two rings or two new copies of this DNA. Right? Now, let's talk about the difference, what's going on in eukaryotes. So first of all, eukaryotes are more complex. So instead of just having one DNA polymerase, they have three. Now, the one that I want you to focus on is DNA polymerase 3. This is the one that's doing the actual copying, the elongation of the DNA. Right? The other DNA polymerases are important. DNA polymerase 2 is important for DNA repair, which we'll talk about at the end of this lecture. And DNA polymerase 1 is important to remove primers on the lagging strand. We'll talk about what it means by lagging strand in a little bit. All right, so the one that I want you to focus on is DNA polymerase 3. Now, we have more than just the polymerase and the primers occurring at this site of replication, right? We have enzymes called helicases. Helicases are what help to actually pull apart that DNA duplex so that the polymerase can come in and read the sequence, right? We also have single-strand binding proteins. These help to keep the DNA in its separated form because it wants to just come back together again, right? So the helicase initially separates the DNA, unwinds it, and then the single-strand binding proteins come in and keep it kept apart. And then we have this enzyme called gyrase, DNA gyrase, and this is pretty important because we're creating a lot of tension on either side of this, what they call a replication fork or replication bubble. So in this top picture, you can imagine that the more you extend, unfold, or unwind this DNA as you go, make it bigger and bigger, that's going to cause more and more stress, torsion, right? More twist, inducing more twists in this DNA. And it's going to snap eventually. It doesn't like that. Right? It's not good physics-wise. And so what this DNA gyrase does is it helps to relieve the stress of that twisting. It kind of takes the DNA and unwinds it a little bit. It's not important that you know how it works, just that it's helping to relieve that stress, preventing what's called supercoiling of the DNA. Right? So I mentioned something about lagging strands. I want to talk about that. So let's do a little problem. Let's try to draw out how this DNA would look if we unwound it and we started to do DNA synthesis on both strands once they've been separated. All right, so I'm going to draw over here. We've got our original DNA duplex. All right, and we can distinguish the two strands by identifying the 5' and the 3' end. So on this strand, we know that if this end is 5', that means that the other end here is 3'. And if this end is 3', the other end is 5'. See how they are anti-parallel, 5 pairs with 3, right? Now let's unwind this DNA a bit. Okay, remember what enzyme and proteins are involved in unwinding the DNA? And we've got our labels on here, right? Now let's draw our new DNA. First of all, let's do this one on the bottom. In what direction is new DNA made? The 5' to 3' end, right? So the new DNA has to be complementary. So is the new DNA going to start over here or over here? Let's think about that. Let's just draw a complementary strand here. Which end of this new DNA is the 5' end, and which end of the new DNA is the 3' end? Due to that anti-parallel rule I was telling you about, this is the 5' end and this is the 3' end. So in what direction is our polymerase? I'm just going to draw a little polymerase guy running. Right, he is going in that direction. Right. Now let's look at our other strand. Right, which end is the 3' end and which end is the 5' end? Right, due to our anti-parallel rules. Right, so we have a second polymerase that's actually functioning on this strand as well. What direction is this one moving? 5' to 3' direction of the new DNA. All right, so far so good. Now, for simplicity's sake, I haven't talked about primers, but before these polymerases could come in, they would have to have a primer be placed right here and a primer, I'll just draw kind of like that, and a primer placed here, right? But just for simplicity, I'm ignoring primers right now. Now let's unwind this DNA a little bit more. Again, this is our 5' end, 3' end. Let's draw our DNA that we already copied. All right. So the DNA is unwinding in this left to right kind of direction. Our polymerase that we had here will just continue to go in this direction to continue to make more DNA in that 5' to 3' direction. But it's different when you look at this opposite strand, right? This is the 5' end here, right, and the 3' end here. So what this polymerase has to do is it was here, right? It has to let go and jump backwards and then run and to synthesize the DNA in the 5' to 3' direction. It's kind of crazy, right? So what that means is that this new DNA is a nice continuous new strand, and this one is made up of a lot of little pieces that are made as the helicase unwinds the DNA further and further. And so this is called the leading strand, this new one, and this is called the lagging strand. And it's lagging because it's taking more time, because an enzyme, the DNA polymerase, I think we said 1, has to come in later and put these strands, connect those pieces together, ligate them together. And so this one's always saying, wait for me, because it's kind of having to run, almost like run backwards in a way, right? Now these fragments here that have to be put together and then are made a little bit at a time, those are called the Okazaki fragments, all right? So if you've ever met Dr. Okazaki in the zoology department, he is a retired faculty. You can ask him if he was the discoverer of the Okazaki fragments. I'll give you a hint. But he would still find it a fun conversation. I'm sure he would have lots to tell you about. All right. So here we have a nicer drawing of what I made, but sometimes I think it helps to see things in movement, all right? So we can see on the left here that the DNA, the helix, was pulled apart, and on the leading strand, on both strands, first we had to have an RNA primer put down. Again, that helps the polymerase to know where to start. And then the polymerase comes in and is making new DNA in the 5' to 3' direction. So as this helix gets opened further, the polymerase on the leading strand just keeps going. It's a continuous strand. Whereas on the other side, the DNA polymerase goes in the 5' to 3' directions, so it's running away from what we call here the replication fork. As more DNA is synthesized or is separated, it has to hop back and make more. And so we've got these little pieces that are being made at a time. It's called discontinuous, this lagging strand, and another polymerase has to come in and connect these pieces later. Again, I just wanted to point out that where the DNA is opened up, where the helix is separated, is called the replication fork. Some other details, we talked about how this primer needs to be put down that's made out of RNA. The enzyme that does that is called a primase, right? It makes the primer. Again, we said that the lagging strand is semi-discontinuous, and that is because the DNA polymerase can only run in one direction. Remember what it is? From the 5' to the 3' end. And again, those fragments of DNA on the lagging strand are called Okazaki fragments. All right, so some other details. We have an enzyme called DNA polymerase 3, right? This is the enzyme that we talked about that is doing the synthesis. We have a part of it, a subunit, that is called the sliding clamp. And what this does is this helps the DNA polymerase from falling off. And so it just keeps it from doing its job. It's called processivity, basically how long the polymerase stays on the DNA as it's copying it before it lets go. So the subunit, this beta subunit of the polymerase acts as a clamp, or a sliding clamp, to encourage processivity. Now, we talked about having this Okazaki strand, Okazaki fragment, right? We also talked about having primers, right? So we've got these primers that have to be taken care of, and then we also have these gaps, right, between these DNA fragments. And so we have the primase, right, that made the primers. We then have a DNA polymerase 1 that comes in and removes those RNAs and replaces them with DNA. And then we have an enzyme called ligase that helps to seal the backbone. It connects those two fragments together, creating a phosphodiester bond there, and it's ligating. Or if you've ever heard of the word ligate, it's kind of like to glue or put together, so that's why it's called a DNA ligase. Now, eventually, as we said before, right, the DNA is all copied and it has to stop. And the polymerase lets go. And also that DNA gyrase that we talked about before, it helps to separate the new and the two copies. Okay. Now, in prokaryotes, we talked about the replosome. All right, so this is, there's going to be two of them, right, that go in opposite directions on that ring of DNA. And the replosome contains a primosome, so that's going to be the primase that puts down the primer, the helicase that separates the duplex and other accessory proteins, and then we also have the DNA polymerase II as part of this replosome. Let's look at the parts in more detail. So you should be able to identify all of these parts and know what they do. Here we have a gyrase, right? This is what's going to help as this DNA gets separated to relieve this tension to prevent supercoiling of DNA. We've got our helicase that actually separated the duplex, and we've got our single-stranded binding proteins that are keeping the DNA pulled apart. You can see on the leading strand here that we've got all this new DNA shown in blue. We've got our polymerase here, and we've got the sliding clamp that's helping maintain processivity, right? It's helping the polymerase to stay attached to the DNA and not fall off. On the lagging strand, I want to point out here, you can see that here is some pieces of DNA that have already been made, all right? Here we have a DNA polymerase currently running along. We've got a primer that got put down, and we have running from the 5' to the 3' direction. DNA polymerase is making new DNA. It also has its own beta clamp. And we said that these fragments need to be connected, right? And the RNA primers need to be removed. So you can see that the DNA polymerase 1 is doing that, right? DNA polymerase 3 is the one that is synthesizing the new DNA. DNA polymerase 1 is the one that's removing the RNA, and it's replacing it with DNA, and then the ligase is connecting those two fragments. Let's see, anything else that I missed? Here we've got the primase. So we've got a new region that's ready to be copied. At the 5' end, we've got a primase putting down a primer. In a minute, this polymerase 3 is going to let go. It's going to jump over here and run and synthesize DNA in the 5' to 3' direction. This clamp loader in the center that looks most alien-like is going to be helping with the beta clamp, the DNA polymerase, repositioning it on the lagging strand. Now we're going to watch a video because, as I said, watching things in movement, especially when the process that you're trying to study is a moving dynamic process, can be really helpful. And also with this video, I just love the detail of it. They try to make all the molecules look realistic. So let's get this moving. Now, I figured this would be an issue, so you can watch these videos on your own. I encourage you, do not skip this. All right? It's really important for you to understand how this works step by step and all the parts. So check out these two videos. I'm going to go through kind of the steps of DNA replication in these images, but again, it's really helpful to watch a video as well. So here we have some DNA that's been separated, and you can see that on the leading strand, the DNA polymerase 3 has generated new DNA in the 5' to 3' direction. It runs towards the replication fork. Whereas on the lagging strand, because DNA is anti-parallel, and the polymerase only runs in one direction, as it runs in the 5' to 3' direction, that means it's running away from the replication fork. And so for both the leading and the lagging strand, the polymerases first need to identify where to copy the DNA by finding an RNA primer. And this RNA primer is put down by a primase. The DNA has previously been separated by a helicase enzyme, and single-stranded binding proteins are going to help keep the duplex apart. The polymerase finds the RNA primer that's been put down by the primase, that's where it starts, and it runs generating new DNA in the 5' to 3' direction. It is attached, or it's associating with the DNA with the help of the beta clamp, right? Again, this is the DNA polymerase 3. And so the leading strand polymerase is just going to keep going. As soon as more of the DNA gets separated, it's going to keep moving forward. With the DNA polymerase on the lagging strand, it's going to run until it hits previously made fragment. And it's going to let go and move backwards towards the replication fork to find a new primer. So here we can see this polymerase on the lagging strand is finishing up. More of the DNA has been separated. The primase has put down that primer region for the next fragment. And so once it finishes that fragment, the polymerase is going to let go, right? And the lagging strand now has the opportunity for the DNA polymerase 1 to come in to replace the primers from previous fragments. And now this DNA polymerase is going to move to the next available region. Meanwhile, the DNA polymerase on the leading strand just keeps chugging along. Here you can see the clamp loader has helped move the beta clamp as well as the DNA polymerase to help get it in this new spot, this new available region, specifically where that RNA primer is. And now the DNA polymerase on the lagging strand can start making more DNA. Meanwhile, previously made DNA, the primer has been replaced with DNA. Remember, the primer is made out of RNA originally. And then the fragments are patched together with DNA ligase. And now we've got this DNA polymerase on the lagging strand is in its new spot, and it can start synthesizing more DNA. Meanwhile, the gyrase, don't forget, has been working to prevent supercoiling as this DNA duplex has been pulled apart. Now, let's talk more about eukaryotic replication versus prokaryotic replication, right? So one of the big differences between eukaryotes and prokaryotes is that eukaryotes have a lot more DNA. Not only do they have more DNA, they have a linear structure, right? It's just one super long strand. And so again, prokaryotes, a lot less DNA, it's just one ring structure. And eukaryotes, there's more of it, and it has this linear structure. And so the basic enzyme functions, like you need a polymerase, you need a primer, you need a helicase, all that is the same. But what is different is what we do when we get to the ends, when the DNA needs to stop. Now with prokaryote replication, the replisomes, they run in opposite directions, and once they meet each other, that termination site, they let go. But with DNA, they don't meet each other. They just go in opposite directions until they fall off the DNA, right? Because it has an end. So also because there is so much DNA, there's not just one replication origin. You can see in this cartoon that you can actually have along the length of the DNA multiple replication bubbles or replication forks, and the DNA would just be synthesized out in each direction until it finds newly made DNA by another replisome, and then it just lets go. So there's multiple replicons, multiple sites, origins of replication to help speed up copying of DNA since there's so much of it. There's also going to be more factors involved in forming this replisome, right? So just to help with initiation of the DNA replication complex. And the DNA polymerase itself is going to be more complicated. It's going to have more subunits to it. Now we have this problem because our DNA is linear that I want to point out, right? So what I want you to notice here on the top right, we have DNA that has been copied, right? So our original DNA is shown in green, and our new DNA is shown in blue. Now on the lagging strand and on the leading strand, we talked about how primases put down a primer, and that primer has to be replaced because it's made of RNA with DNA, right? So this gets removed, right? This primer gets removed, a DNA polymerase comes in, and what it does is when it comes in, it identifies where the gap is. It finds this 3' end of this fragment, and it says, oh, this is where I need to work, all right? And it comes in and it replaces that primer right there. But at the very end of a DNA, there's nothing next to it right here on this side for the polymerase to identify. And so what happens is that the primer gets removed, but it never gets replaced with DNA. And so you just have this piece hanging off the end, and as DNA gets copied, it gets lost. And so what happens is the DNA gets shorter and shorter and shorter, and that would not be good, right? And so these regions of the chromosome, the very ends, are called telomeres, and eukaryotes have a mechanism to prevent the DNA from just getting shorter and shorter. So a telomerase comes in and basically it makes some extra DNA on the end so that when the polymerase comes by, it can identify where that gap is that needs to be made. And this extra DNA on the end, it's not coding for anything important, so if that gets cut off, no big deal. Right? So the enzyme is called the telomerase, and the region of DNA that it extends, that it adds on, this extra DNA is called the telomere. Right? And now when that primer gets removed, the polymerase comes in, it sees this DNA there, and it's like, oh, I can see the gap now. I know where to work, and it'll copy that important DNA, and it won't get shorter and shorter. And so scientists think that the regulation of how long our chromosomes are and the maintenance of these telomeres is correlated with aging. And similarly, cancer cells, which seem to never die or never age, they seem to have more activation of this telomerase enzyme. It's a really interesting. Now, here's another video that's probably not going to run. But I encourage you, this is going to talk about telomeres, what we call this end replication problem, and how telomerases can fix it. All right. Let's talk about our final objective, mechanisms of DNA repair. Now, we know that mutations occur, right? But what are some ways that mutations can happen, right? So first of all, we know, if you're exposed to the sun, it's UV light, it's bad for your DNA, it can cause DNA damage. But also, every time that DNA gets copied, that polymerase has to work perfectly, right? Remember, A binds with T and C binds with G. What if something happens where it accidentally puts down a C instead of a G or a T instead of an A, right? That is a mutation, right? And so DNA has to be copied perfectly, but we know that we live in an imperfect world, right? And so even though they are very, very good enzymes, and sometimes they make mistakes, we do have proofreading functions of these enzymes. They go back and they double check, but even with that, they make mistakes. And so we have Anytime a cell divides, there's an opportunity for a mutation, and then environmental exposures are also an opportunity for DNA damage to occur. And we know that DNA repair is really important because there are multiple levels of systems and also mechanisms that check to see whether or not DNA it has been damaged or not. And so there's two general categories of DNA repair, specific and non-specific, as you can guess, right? There's some DNA repair systems where they only fix one type of damage. And then there's others that are more multi-purpose that will come in and fix a variety of different types of problems. So let's look at one example of a specific repair mechanism. So we talked about UV damage, right? What actually happens when there is UV damage? So on the right here, you can see a sequence of DNA, and it has two thymines next to it. That's fine, right? But what UV light can do is it can cause this thymine dimer. So it'll kind of mess with the structure of the DNA there. See how it's not base pairing with its complement anymore? Now they're connected to each other. It's got this weird bump in the DNA. And these thymine dimers are kind of hard to copy correctly. And so when that DNA gets duplicated, it may not correctly copy that there's two T's in a row. So there's an enzyme called photolyase. So this DNA repair enzyme, this is the only type of problem it fixes. It's a specific repair mechanism. It comes in and it will cleave that dimer and help to get those thymines back where they need to be in the correct structure. Now, a non-specific repair mechanism is called excision repair. It's kind of like surgery, right? It goes in, it finds an area where there's a mutation, it cuts out that DNA and some DNA around it, and then new DNA is made to fill in that gap. So we have three parts. First, recognizing where the damage is. Second, removal, excision of the damaged DNA. And then thirdly, recreating, putting down new DNA that is the correct sequence, right? You don't need to memorize, but the complex that's involved is UVR, ABC, right? Don't need to memorize that. But it's called excision repair and it's non-specific, right? And that's what you should know. Now, for this chapter, there is an assignment associated with it. So I encourage you, if you get stuck on any problems, to go back to these slides because we talk a lot about the concepts in that worksheet. Also, that worksheet is going to be a great study aid for the mini exam. Have a great day.